All right, on to vertebrates. And so I think you can figure out that I'm drawing a fish for you here, and we're going to fill in with the circulatory system, the closed circulatory system of a vertebrate. And so we'll start with the ventricle, which is a muscular pump. It pushes the blood forward through this thing we saw in the laboratory. It's called the bulbous arteriosus, and it connects to the ventral aorta, which then pushes the blood through gill arches. Some on the right side, some on the left side of the fish. I'm showing you the left side ones here. One, two, three, four. One, two, three, four. There might be as many as six or seven, depending on what kind of fish you're talking about. Okay. And then at the very top, the gill arches convene at the dorsal aorta. The blood pushes blood forward to the head, backward to the rest of the body. And I'm going to show you the part of the circulation that goes back, back, back to the tail of the fish. And then we've got the venous return of the blood back to the front part of the fish. It's eventually going to go back into the heart. But to get to the veins, the blood has to pass through capillaries. The capillaries are running from the arteries to the veins, and then by the time the blood gets to the front of the fish, it goes into this general membrane-bound sac, the sinus venosus, and then from the sinus venosus, the blood passes into the atrium and then into the ventricle. So I'm going to point out a couple of parts of the circulatory system that are of interest, starting with the ventricle. The ventricle is the real powerhouse of the circulatory system. It pushes the blood forward into the bulbous arteriosus. Okay, now the ventricle is considered part of the heart. Bulbous arteriosus is not. Why not? Well, the bulbous is not a muscular pump. It doesn't actually have any mechanism to physically create pressure. It's a passive pump. I mean, it's got the elastic walls, it's got the compliance that allows it to expand and apply pressure, but that's basically the residual pressure that's created from the ventricle. Okay, the bulbous arteriosus, as you might remember, it's the sac that allows for blood to flow more or less continuously through the gill arches because of its elasticity. The ventricle pumps and stops, pumps and stops, and to generate a continual amount of positive pressure through the gill arches, we have to have that bulbous arteriosus. Okay. Now the other part of the heart is the atrium. Okay, I don't know if you could see it there. I put an A there, but the atrium is really important because it assists the filling of the ventricle. The atrium is muscly. It's not nearly as muscly as the ventricle. The ventricle has to create the pressure to pretty much power the flow of blood throughout the entire closed circulatory system. Okay? The atrium simply has to assist in the filling of the ventricle. Okay? Without the atrium, the ventricle is simply filling passively, and a relaxed muscle, a relaxed ventricle, is not going to fill as effectively as it would if there were an atrium behind it to contract and fill the ventricle when the ventricle is in the state of relaxation. Okay? The other thing that we have is the sinus venosus. The sinus venosus is definitely not a muscular pump. It simply receives the blood from all different parts of the venous return. And that's a closed circulatory system with a two-chambered heart that we find typically for fishes. Now, the other salient part to this anatomical lesson is the swim bladder. Now, the swim bladder is a buoyancy organ. We see it evolving probably in the Devonian and the bony fishes by having this gas-filled sac inside the fish. It's able to maintain neutral buoyancy if the volume of air in the gas bladder is adequate to compensate for the otherwise negative buoyancy of the fish's tissues. Now, the basic anatomy that I've drawn here is one that's called physostomus. Physostomus anatomy of the swim bladder. And by this, I mean that the swim bladder is actually connected to the digestive tract. Okay, so here's the cardiac stomach. Here's the tube that connects the cardiac stomach to the pyloric stomach. The pyloric stomach is going to be attached to the intestines. You get that part. Here's the esophagus. A physostomus swim bladder is actually physically attached to the GI tract. Okay? And in this situation, the mechanism by which the fish is filling or deflating the swim bladder is probably going to involve going up to the surface, gulping air, pushing that air into the swim bladder, or burping the air out if the fish needs to increase its overall body density. Okay? Taxonomically speaking, we associate physostoma swim bladders with those fishes that are kind of basal. The non-acanthamorph fish, like the sardines, the trouts, minnows, which include the carps, in the catfishes. There are actually several common types of fish that we find, many of them in fresh water, that are not in the acanthamorph group. Now, we talked about the swim bladder in the context of adaptations to life in the Devonian, which were flooded inland seas that were seasonal. They had a tendency to dry up, and during the dry season, the animals needed to make greater and greater use of terrestrial habitats. The other thing that happens as these ponds of the inland seas are drying up is that the oxygen content, the dissolved oxygen in the water goes way, way down. Okay, now warm water tends to hold less oxygen than colder water. If you have any kind of biological oxygen demand, if the water is dirty or if there are bacteria or algae decaying in the water, all of that activity is going to result in the depletion of oxygen. Okay, so fishes that are still living in the water under those conditions are going to be subject to relatively oxygen poor conditions, and it might reach the point where the ability of fishes to take up oxygen through their gills in this area here is going to be compromised, right? Now, thinking about it, this physostoma swim bladder that we have in the fishes lends itself as a potential air-filled space. We're going to be able to breathe in air which has got a pretty high concentration of oxygen, which has got a pretty high percentage of oxygen. And the surface areas of the swim bladder might be a great place to supplement the amount of oxygen that's taken in through the gills. I mean, if the water isn't cutting it, if the water isn't providing adequate oxygen to meet the fish's metabolic needs, why not use the air? We've got quite a bit of oxygen in the atmosphere. We've got those plants doing their oxygenic photosynthetic thing. And so when the oxygen level drops, we might be thinking about using that swim bladder as an additional source of oxygen. Okay? And so imagine, if you will, oxygen depleted blood supply to the surfaces of the swim bladder. Now, as the blood is passing close to that surface in the swim bladder, it's able to pick up oxygen from the swim bladder content. And then when the veins leave the swim bladder heading back to the heart, that blood could be effectively fully oxygenated. All right? What I'm saying here is that the physostomous swim bladder is serving as a functional lung. And not only is it a functional lung, it's actually homologous with a lung. And the pulmonary arteries that we have in terrestrial vertebrates actually does derive embryonically from a sixth gill arch. Okay? A sixth gill arch that goes in the ventral aorta. Instead of heading to the dorsal aorta, it goes to the lungs. Basically, it creates the pulmonary arteries. And so again, not only is this physostomous swim bladder functionally a lung, it's actually homologous with a lung. The physostomous swim bladders of modern fishes like trout are deriving from the same ancestral structure that gave rise to the lungs in terrestrial vertebrates. Now, the second thing to note here is that this mechanism of physostomous swim bladders, of having to go up to the surface to fill your swim bladder with gas and then burping it out as you need to dive deeper, this is not really a viable option for fishes that maybe have gone back to saltwater and are living full time in really, really deep water, right? A fish that lives at 300 meters below the surface is not going to be able to swim to the surface, gulp air every time that it needs to adjust its level in the water column. Okay? And so when we look in maybe the majority of saltwater fishes, like probably many of the ones we dissected in our fishes laboratory, we have a different anatomy. This kind of anatomy is called physoclistus. And the physoclistus swim bladder has no connection whatsoever with the GI tract. Okay? Now, fishes with physoclistus swim bladders would not be living under conditions where they need to use their swim bladders as a supplement for oxygen. They live in water that's not anoxic. And so the blood that comes away from the gill arches is fully oxygenated. This O2 rich blood supplies a gas gland which is capable of it actually uses a countercurrent mechanism to increase the levels of blood so great that it starts to secrete oxygen gas into the swim bladder. Okay? And that's how the fish is able to increase the total volume of gases. If it needs to raise in the water column, it could basically take oxygen out of its blood, put it into the swim bladder, and then increase its overall buoyancy. Okay? If it needs to decrease buoyancy, it can simply reverse that process. It can put additional oxygen back into the blood, and that blood will return back to the heart fully oxygenated, but it's also the mechanism by which we decrease the total volume of gases in the swim bladder. What kind of fishes have physoclistus swim bladders? Well, these tend to be the more derived fishes, including most of the acanthamorpha. So at this point, you might want to visualize the idea that this physostoma swim bladder is kind of like the starting point for the evolution of three different kinds of structures. Okay, first of all, we still have plenty of fishes today that use physostoma swim bladders, and these are the things like trout and minnows and catfishes. Okay, then we have a hugely successful group of very derived fishes that use physoclistus swim bladders, and that structure, from an anatomical standpoint, is really pretty astounding. We have this gas gland. It uses a sophisticated countercurrent mechanism to secrete and absorb oxygen from an airspace inside. That structure is derived from the physostoma swim bladder. Okay? And the third thing we find evolving as a derived state from the fish's physostoma swim bladder are the lungs of tetrapods. Okay? Now think about the origin of this pulmonary circuit, pulmonary circulation, right? Um, So if here's the ventricle and the gill arches, the normal gill arches of a fish, and so we've got an atrium and the sinus venosus, and the sinus venosus is receiving blood from all different parts of the fish. Okay, the pulmonary artery, as we said before, is deriving from one of the gill arches. One of these gill arches carrying deoxygenated blood is going out to a brand new structure. This is the physostoma swim bladder. Right? And we'll be using the inside wall of the swim bladder as a surface for gas exchange, and the capillaries returning blood from the wall of that swim bladder are going to be coming back with a full amount of oxygen, and it's going to be returning back to the heart. Okay? And maybe the only difference that we have in a three-chambered heart system is, of course, the third chamber, and that's going to be the left atrium. Okay? Uh, the right atrium, the one that's still in gray, will still be receiving blood from the sinus venosus, which takes blood from all over, all different parts of the body. Will be sending blood back to the sinus venosus and ultimately to the right atrium. Okay? The left atrium is more specific. It only receives blood back from the pulmonary vein. Okay? And this is, in effect, the same thing as the three-chambered heart that we find in the basal tetrapods, including the amphibians, as well as several of the squamates. So what I want to do now is to redraw this three-chambered heart system, but in the normal way that you'll see in textbooks. Like if you open up the chest cavity of a frog that's lying back down in your dissecting pan, what you'll see are three chambers, and they're laid out kind of like this. Okay, it's kind of an art lesson here. So we'll start with the ventricle of this froggy. Ventricle is a pretty muscular pump, and we'll have a couple of large blood vessels leading away from the ventricle. First, you might sort of visualize a main aorta through which the blood is being supplied to the entire body of the frog. So this is going to be the systemic artery that's kind of like the bulbous arteriosus. Blood is going to be going through this systemic aorta, serving all parts of the animal except for the lungs. Okay, the other blood vessel that's leaving the heart, okay, the other blood vessel is going to be going to the right and the left lung. This is the pulmonary circuit. These are the arteries that go out to the lungs. Now, the blood coming back from the lungs, ultimately the blood leaves the lungs fully charged with oxygen, but it's going to be coming back to the left side of the heart, and it'll be entering into the left atrium. Well, blood coming into the left atrium from the lungs, from the right lung and the left lung. Sometimes there'll actually be a separate pulmonary vein coming in through two openings into the left atrium. Okay. Now, blood from the systemic circuit is going to go, as I said, to all different parts of the body. Ultimately, comes back through two different sides. We'll have some blood coming back from the top, from the head portion of the frog, blood coming back from the bottom portion of the frog. So we'll have this inferior vena cava and a superior vena cava, and they basically are going to be putting blood into the right atrium. In this area over here, this is going to be the right atrium on the right side of the heart. The right side of the heart is on the left side of the diagram because we're looking at the heart from the front. Okay, so this is the heart. This is the right-hand side of the frog. This is the left-hand side of the frog. And that's basically the anatomy of our three-chambered heart. It's basically the same thing that we have drawn up here. You can sort of visualize in the color scheme. Blood coming back from the pulmonary circuit is going to be fully oxygenated It's going to be bringing blood, oxygen-rich blood in to the left atrium. Left atrium is going to fill with oxygen-rich blood, and the oxygen-rich blood from the left atrium is going to be pumped into the ventricle. Now, blood coming back from the systemic circuit is presumably going to be oxygen depleted. Okay, so the blood coming back in either from the head, from the superior vena cava, or from the tail and the legs area, it's going to be coming back to the right atrium, and the oxygen poor blood is going to be pumping back into the chamber, into the ventricle from this side. Okay. Now you might note that the right atrium is basically very similar to the normal atrium that we have up here. It's basically receiving blood from all different parts of the animal. The right atrium kind of is the ancestral one. It was the one that we find in most two-chambered heart fishes. It receives blood from all over the fish. The right atrium in our tetrapods also receives blood from all over the body. It's clearly the part that's homologous to the atrium that we find in most fishes. Right? The left atrium is a new structure. The left atrium is something that has to appear for the first time as we make the transition from a two-chambered heart into a three-chambered heart. Okay? One thing that every beginning biology student notes is that because we've got a common ventricle, the blood is going to be mixing. The oxygenated blood from the systemic circuit and the oxygenated blood coming back from the pulmonary circuit is going to be mixing in this common ventricle. So the blood that's going out to the systemic circuit is not going to be fully oxygenated. That would be the blood coming back from the left atrium. Okay? But it also gets mixed with blood that's coming back from the systemic circuit. And you might say, well, that's suboptimal. That's not what we want. I think I would much rather have the blood from the left atrium going exclusively to the systemic circuit, and the blood coming back from the systemic circuit, the deoxygenated blood, going exclusively to the pulmonary circuit. And that's simply not what we have in the case of your typical three-chambered heart. And yet, speaking as a warm-blooded primate, this does appear like a pretty poor design. Okay? If I were born with a frog heart, I would expire instantly. Okay. And yeah, this is actually the starting point for the evolution of a supercharged four-chambered heart, which we'll draw over here on the right-hand side. Okay, so let's start by drawing the ventricle chamber, the common ventricle. Now, on the left side of the ventricle, we tend to have a thicker wall. On the right side of the ventricle, we tend to have a thinner wall. Okay, and from the left side of the ventricle, we still have that systemic arterial system. Okay, this, in the case of a human, would be the dorsal aorta. It would actually make a it would make an arch, and it would basically go back this direction. But for now, let's just call this our systemic circuit. This is the one that's going to be taking blood to all different parts of the body. Okay. The other side of the heart, the right side of the heart, will still be sending blood to the pulmonary circuit, which is going to return the blood fully oxygenated over to the left atrium. So here's our left atrium over here. Oops, I'm doing this in red. Sorry, but it's going to be in red. Okay. Might have two inflows into the left atrium. Blood is going to be coming in into the two openings from the lungs, and basically this left atrium is going to be filling with fully oxygenated blood. Okay. The deoxygenated blood from the systemic circuit, coming either from the head or from the lower half of the body, is coming in through this inferior vena cava, and the inferior vena cava is going to be hooking up to the right atrium. And so the right atrium will be receiving blood that is deoxygenated coming back from the systemic circuit. Okay. Now I've done things deliberately so it actually shows you the similarity that we have between the three-chambered heart and the four-chambered heart. The only difference here is the presence of a septum. Remember that term septum from our discussion of the septated coelom. Okay. And this ventricular septum divides the ventricle into a left ventricle and a right ventricle. In other words, that's where we end up with a four-chambered heart. Okay? And the ventricular septum is a really great idea because it allows us to isolate the oxygenated blood coming back from the pulmonary circuit. And that oxygen-rich blood is only going to be passing exclusively into the systemic circuit. Okay? It's exactly what we want. And on the right side of the heart, we've got the deoxygenated blood coming back from the systemic circuit, and it's going to be pumped by the ventricle only to the pulmonary circuit. Okay? In other words, we're able to isolate the deoxygenated blood, send it entirely to the pulmonary circuit, whereas the returning blood from the pulmonary veins is able to be isolated from the deoxygenated blood, and it goes exclusively to the systemic circuit. Okay? Now I got to admit, that's a really great circulatory system, but it's really not that different from what we had in the case of a frog. Okay? The only difference is the presence of this ventricular septum. Now think about it. What would it take for an animal with a three-chambered heart, like the one that we have over here, okay, to increase its metabolic performance? What could be done to even create a marginal amount of isolation between the right side of the heart and the left side of the heart? Okay. Would creating a little speed bump, maybe a partial ventricular septum like this, like right there? You see that? Put a partial ventricular septum in right there at the bottom of the heart. If we have a little speed bump there, that would have the effect of tending to isolate the flows, keeping the oxygenated blood on the left side of the heart and the deoxygenated blood on the right side of the heart. And you could say that a partial ventricular septum is better than no septum at all. Okay, um, if if animals were under selection for increased metabolic performance, animals that had a partial ventricular septum, a partial isolation of the blood in the ventricular chamber, is going to have a distinct advantage relative to one that has no septum at all. Okay? And an animal that has a slightly bigger ventricular septum is going to be at an even greater advantage. And so you could actually, parse this out into 10,000 tiny steps. You could see that over the course of gradual evolution, you could start with no ventricular septum whatsoever and end up with a complete ventricular septum with a fully four-chambered heart. And that could be easily explained by selection being driven by a demand for greater metabolic performance. Okay, now what kind of animals have four-chambered hearts? Well, mammals and birds, obviously, but crocodilians, alligators, gharials, crocodiles, they also have four-chambered hearts. Okay. What kind of animals have three-chambered hearts? I think we talked about this before, but let me list them anyways. You've got all the amphibians, you've got turtles, and you've got squamates, lizards and snakes. And in the case of squamates, you've got partial septa. Those lizards, especially the lizards that have the highest metabolic performance capacity, are often the ones that have the greatest degree of partial septation of their ventricles. Now, what about the dinosaurs? And by this I mean the non-avian dinosaurs, because obviously birds have four-chambered hearts. What does the existence of four-chambered heart anatomy in both birds and crocodilians suggest about the character state for all of the dinosauria? Okay. Now, you might have had to refer back to your notes, but you might remember that the Archosauria consists of actually three or four groups. The most derived of the archosaurs are the pterosaurs, crocodiles, and the dinosaurs. Dinosauria includes the birds. We're saying that this character state of having four-chambered hearts is present in the aves, it's present in the crocodilia. I would presumably guess that the pterosaurs would probably also have had four-chambered hearts. Okay, I think it's a pretty good guess because they're capable of powered flight. Now, if this is the case, it would make sense for the Archosaurian ancestor to also be an animal that had four-chambered hearts, which would suggest that all of the dinosauria, including all of the ornithischia as well as the theropods, those are the ones that include the aves, would likely have had four-chambered hearts themselves. Now, there's another interesting twist when it comes to crocodiles. Crocodiles are able to facultatively shut down their pulmonary circuit. And this makes sense when the animal goes on these long submersed periods. It can hold its breath for several minutes at a time. And during that time when it's submersed, it's not really getting any bang for its buck from its pulmonary circulation. So what it can do is to redirect the blood from the pulmonary artery over into the systemic artery. There's a little shunt. It's called the foramen of Panizza. And the foramen of Panizza becomes open. It opens up when a crocodile goes on a dive and stays underwater for a long period of time. And when the crocodile surfaces again, starts breathing air, it'll shut down the foramen of Panizza and the blood will once again circulate out to the pulmonary circuit. You can sort of see this here. Neither of these diagrams is as good as mine, but basically the foramen of Panizza is here, connecting the arterial systems coming from the right ventricle to the left ventricle. Here you can see the foramen of Panizza connecting the pulmonary artery to the systemic circuit. Okay? Now this might be sort of just a little bit of crocodile trivia, but it's actually kind of similar to something that goes on in the circulation of humans. Okay? Because as fetuses, we have maybe not a foramen of Panizza, but we've got a foramen ovale. Okay? foramen ovale is doing effectively the same thing because when we're fetuses, we don't use our lungs to acquire oxygen. All of our oxygen is coming from the systemic circuit, the part of the blood that goes out to mama's placenta is where we get both nutrition as well as the respiratory gases. The blood still circulates out through the pulmonary circuit to the lungs, but that's mostly for like practice. Um, Baby needs to practice circulating blood out to its lungs before it's born, I guess. But we can make better use of baby circulation if we were to concentrate most of our circulation out to its systemic circuit. Going back to our picture here is that the blood is going to be passing from this area over here through the foramen ovale over into the left atrium. And that's going to reduce the overall volume of blood being pumped out of the right ventricle into the pulmonary circuit. Another truly Bio 202 type of issue is the question of where do hearts come from? What's the origin of the heart in animals? Okay. And part of this answers come in the last 30 years or so as a result of some really interesting studies done by a couple of fruit fly geneticists working in Europe. What they did was they isolated mutations of fruit flies that caused baby fruit flies, the maggots, to die very young. And they're able to study these mutations in great detail. And one of the mutations that they found was a mutation that caused a little maggot to develop that didn't have any heart whatsoever. Okay? There was a heartless maggot. There were no steps even beginning to start in the formation of a heart. It's a completely lethal mutation. And our geneticists gave the name for this mutation Tin Man, after the Tin Woodsman in the Wizard of Oz. Okay, for those of you who don't know, he was the one that didn't have a heart. He was going to see the wizard because he wanted the wizard to give him a heart. Okay, anyways, Tin Man was really exciting because it's obviously something that's really important. It's crucial for the development of a heart. Now, one thing that we do when we find a really interesting gene like this is to look for the activity of that same gene in other kinds of organisms. Okay, and we do this through a process called in situ hybridization. We can create little tags that would bind to messenger RNA molecules that are being transcribed from a similar gene. A gene that's close enough in sequence to Tin Man would be producing a similar enough RNA molecule that would be able to locate the occurrence of those RNA molecules through this process called in situ hybridization. And what we have on the right-hand side of this picture is an early embryo of a zebrafish. Danio, the zebrafish, the same one that you could find at PetSmart in the aquarium section. Okay, and this zebrafish embryo is showing us that the Tin Man gene from Drosophila seems to be active in this area of the fish's anatomy. Okay, and I don't know if you can see this, but this is the head of the fish. This would be the mouth. This would be the gills. What do we normally find in this region of the fish's anatomy? Right, the heart, right? Exactly. So that's what makes this ridiculously cool is that this Tin Man gene, the gene that is there obviously in both vertebrates, like the zebrafish, as well as in fruit flies, it must be some type of master switch. It's a master switch gene that triggers the development of the heart. Now, the heart of the fruit fly is, at least in the adult organisms, a completely different structure compared to the heart that we find in vertebrates. It's dorsal, it's part of an open circulatory system, and that's completely different from the heart that we find in the case of a zebrafish. Okay? And so at this point, you just say, yeah, I remember that. We talked about this earlier. We said that the presence of a gene that is so clearly homologous between two species means that the most recent common ancestor shared by those two species must have also had that characteristic. In this case, this master switch gene, Tin Man or NKX, would have also been there at the protostomial deuterostomial common ancestor. Now, what about this? Remember nematodes? They don't really have a heart at all. You can say they have kind of a primitive open circulatory system, but they don't have any muscular pump circulating the coelomic fluid. That's something that occurs in more complex open circulatory systems like we have in a fruit fly. Okay? In the case of a nematode like the C. elegans, there's no heart. But when we look at the genome of C. elegans, we actually can find a gene that looks like it's homologous with the NKX Tin Man, the master switch for the heart. It seems to be what we find serving as a master switch for the throat, the development of the throat of C. elegans, and presumably all nematodes. And this actually makes perfect sense. The pharynx of a nematode is also a muscular pump, just like the heart is. Okay, and so you might remember back to the first half of this lecture where we said that um, maybe the heart got its start as a second or ectopically placed pharynx in the body cavity of an ancestral bilaterian. Well, that story is not all that implausible, given the fact that both the pharynx master switch gene and the heart master switch gene appear to be homologous. Okay, I'm going to end this lecture here, and we'll be starting the next time on respiration, respiratory pigments.